Hi, everybody, and thank you so much to the organizer for giving us this space. My name is Sonia Moreno. I'm postdoc at Stanford University, the Department of Biomedical Data Science. But let me highlight that this study was conducted during my stage in Fundación ACE. Today, I would like to tell you about genetic studies in Alzheimer's disease, and specifically about how we can enrich our genetic studies using a specific subset of Alzheimer's disease patients. So I would like to talk first about the heterogeneity that there's inside of Alzheimer's disease. Dr. Viswanathan suggested several years ago that dementia could be understood as a gradient of neurodegenerative but also vascular features. So as you can see in this figure, in the slide, in one side we could have those individuals presenting pure AD forms, and in the other, those individuals presenting pure vascular forms. But in the middle of those, there's a large gradient of AD patients which present mixed pathologies. A mixed pathology, which means that they present at the brain signals or the pathological hallmarks of both the neurodegenerative hallmarks and the vascular too. So in the middle of these both conditions, we are going to find a mix. So considering that, we decide to take into account this in our genetic study. And our objective was trying to assess if across all these different gradients, we were able to identify different genetics and also different pathways conducting to the disease. So for doing that, the first thing that we did was trying to generate a new cohort of Alzheimer's disease patients. This is the Genome Research at Fundación ACE cohort, which includes two phases or two stages. The first one is that included in this study. This present near to 70,000 individuals, and all the cases, which is I think one of the most important pieces here, all the cases were recruited in a single center at Fundación ACE, which means that they were evaluated by the same medical team and using the same procedures. So thanks to that, we were able to generate this gradient proposed by Viswanathan, which covers all the spectrum from the neurodegenerative to the vascular features, and we were able to generate subsets of individuals along of this spectrum. So, and as you can see here, these are the numbers that we were getting for each of the different subgroups that we were able to generate or extract from our cases. So next, what we did was conducting a classical genome-wide association study. We make the genotyping, we make the quality control of our data, and here you can see that our group of cases and controls, that you can find them in the plot in red and in black too. This is a group of individuals that match perfectly with European ancestry, which is expected because all of these individuals are from Spanish ancestry. And here, this is a classical Manhattan plot, which is a way to show our genetic results, coming from a genome-wide association study, and considering our sample size, the unique genome-wide signal that we can find here is ApoE, which is expected considering that ApoE is a strong genetic risk factor for Alzheimer's disease. So after conducting this initial evaluation of our data, the next idea was trying to assess if across all the different subgroups of Alzheimer's disease patients that we were able to generate along of the gradient or the spectrum, we were able also to detect different variants linked or genetic factors linked with Alzheimer's disease that present a differential behavior or effect across the subgroups. So this was our next question. Are load variants differentially associated with a specific subgroup of Alzheimer's disease patients. We evaluate this, and then our objective was trying to evaluate if we were able to identify patterns across these variants, where we could generate subgroups of variants presenting the same behavior. Our next idea was trying to extract correlatory networks coming from these variant subgroups or categories, and then conduct a pathway analysis. So after conducting this analysis, in fact, we were able to identify 3 categories of Alzheimer's disease variants, meaning that we were able to detect category A, or what we call category A, which were those variants who present an effect which increase with AD certainty, or in the same way, which increase if we enrich the subset of Alzheimer's disease patients with those presenting a more pure phenotype of Alzheimer's disease. Then we detect a second category, which we call category B, of variants for those that present an effect size which was decreasing with AD certainty. And finally, we detect a third category where all the effect of the variants was stable across all the subgroups of Alzheimer's disease patients. Then, as I was mentioning before, our objective was trying to detect if these variants were conducting us to different biological pathways related with Alzheimer's disease. And in this table, you can see here the different pathways that we find associated with the variants and with the correlatory networks that we extract for each of the 3 categories. So as you can see here, the immune system is prevalent across all of them, which is expected considering that immune system is one of the top pathways linked with AD. But surprisingly, we find here some new pathways only related with category A. These pathways were the regulation of vasculature development and blood vessel morphogenesis. Then our second approach for analyzing this data was trying to conduct a classical meta-analysis of genetic information. For doing that, or meta-GWAS, but we call this meta-GWAS. So the next thing that we did was trying to merge the results that we were able to obtain in our GRACE study with those that were available in public resources. We used datasets coming from dbGaP, and we also use summary statistics coming from the International Genomic Alzheimer Project. So merging these 2 sets of information, we get the first stage of our meta-analysis, which was the discovery phase. In a second phase, our objective was trying to replicate those signals that were reaching the genome-wide significance at the first stage. After doing that, in a classical meta-analysis, the next step is trying to meta-analyze the discovery and the replication. And this is the protocol that we were following. So after doing this, we were able to identify 2 new genome-wide significant signals. The first signal was located in chromosome 5, specifically in a region that for us was surprising, because in this region you can find here the enzymes that synthesizes cholesterol. So as you can see here, our lead SNP, this is a region which is in high linkage disequilibrium, which means that we are enabled to know which ones of all these genes is the causing one, the causal gene. The second region was located in chromosome 8, and our top signal, which you can find here, was located between these 2 genes. So our study, let me give you three fast highlights or conclusions from our study. The first one is that we were able to detect different gene categories operating differently across AD subgroups. We were able to identify new pathways linked with Alzheimer's disease. These were the vasculature regulation of the vasculature development and blood vessel morphogenesis. And we were able also to detect 2 new genomic regions using a meta-analysis strategy. So please let me remember you that all the genetic data that was generated during this study is available on the European Genome and Phenome Archive. This is the extension number, and you can go there and just download all the genetic information. Please let me thanks you for your attention. And please, if you have any doubts, don't hesitate to contact me. It will be a pleasure for me to answer to any of your questions. Thank you so much.